Hi guys, today we are talking about the structure of DNA. So we're going to get right into it. There's a little bit more vocab with this section. So first is a nucleotide. A nucleotide is a monomer that forms DNA. It's made up of a phosphate group, a sugar, and a nitrogen-containing base. Double helix is a model that compares the structure of DNA molecule in which two strands wind around each other to that of a twisted ladder. Base pairing rules are rules that describe how nucleotides form bonds in DNA. Adenine, A, always binds with thymine, T, and guanine, G, always bonds with cytosine, C. In the 1920s, scientists have known now that DNA was a very long polymer or chain of repeating units. These small units or monomers are called nucleotides. There are 4 separate nucleotides that make up DNA. There are 3 main components to a nucleotide. A phosphate group, which is 1 phosphorus with 4 oxygens. A ring-shaped sugar. In DNA, it's called deoxyribose. And a nitrogen-containing base that is composed of a single or a double ring structure that is built around nitrogen and carbon atoms. Although DNA is billions of nucleotides long, there are only 4 nucleotides that make up the composition of our DNA. The only difference is the type of nitrogen-containing bases that there are. Cytosine and thymine are both single ring structures, and that's why they pair with double ring structures, adenine and guanine. Here we have adenine. Adenine pairs with thymine, double ring and single ring. And then cytosine pairs with guanine, double ring, single ring. In the 1950s, Erwin Chargaff changed the thinking about DNA by analyzing the DNA of several organisms. Remember, in the 1950s, it wasn't fully confirmed that DNA was in fact the genetic material. Our DNA does not have the same amount of each nucleotide in it, and that's something that Erwin Chargaff noted in his study of several organisms. However, he found the same 4 base pairs in all of the DNA of every organism he looked at. But the proportion of the 4 bases differed between organisms. However, his findings were that roughly the amount of A always equals a rough amount of the number T, and the amount of C roughly always equals the amount of G. And this became known as Chargaff's rule. Shortly after Hershey and Chase confirmed that DNA was in fact the genetic material, 2 scientists at Cambridge University in England developed an accurate model of the 3-dimensional shape of DNA. They were supposed to be studying the structure of proteins, but they were more fascinated by the challenge of figuring out the structure of DNA. They were sparked by all the scientists that came before them, specifically Hershey and Chase, Erwin Chargaff, and a scientist by the name of Linus Pauling, who found that the structure of some proteins was a helical structure, or a spiral. There was a lot of competition in the field at the time to discover the structure of DNA. Watson and Crick were supposed to be studying proteins, but they were highly fascinated by the challenge that DNA posed. Rosalind Franklin and Maurice Wilkins were 2 scientists at another university in London. They were studying DNA by using a technique called X-ray crystallography. This bombards DNA with X-rays, and the atoms in DNA diffract into an X-ray pattern that is captured on film. There's then a big mathematical equation to help differentiate what that pattern means. Rosalind Franklin's most famous picture is this one right here. This is called Photo 51. It depicted DNA as an X-shaped structure that was surrounded by a circle. James Watson was an expert in understanding and differentiating X-ray crystallography patterns. So he knew exactly what this structure was by looking at Photo 51. However, he did not obtain Photo 51 in a very appropriate manner. He wanted to work with Rosalind Franklin and Maurice Wilkins, and she said no. So Maurice Wilkins snuck into Rosalind Franklin's lab late one night, took photo 51, and gave it to Watson and Crick. This led Watson and Crick to make a model out of metal and wood to figure out the three-dimensional structure of DNA. Originally, they had placed the sugar phosphate backbone on the inside because that makes sense. The backbone runs through the center of organisms. However, they quickly realized that if they put the sugar phosphate backbone on the outside and the bases in the middle, that it actually led to a way of how DNA can replicate itself. Originally, they thought like would pair with like, A with A and T with T, but realized that that led to an uneven width of the DNA. Photo 51 showed a very uniform width and quickly decided to pair a single ring structure with a double ring structure. That gave the helix an even width throughout. In 1953, Watson and Crick showed their findings in the journal Nature. It showed that DNA was a helical model in which two strands of DNA wind around each other like a twisted ladder. They also stated that the strands were complementary, that they fit together and are the opposite of one another, which led to an understanding of how DNA can replicate itself. One thing that their article did state is that all nucleotides of a single strand are joined together by covalent bonds in the backbone that connect the sugar of one nucleotide to the phosphate of the next. The backbone is made up of alternating sugar phosphate backbone or bonds on one side of the double helix. The two helixes are held together by hydrogen bonds between the bases in the middle. Alone, hydrogen bonds are very weak, but when they are strung together as DNA does, it holds DNA together very strongly. The base pairing rules are strands of DNA always pair up in the same way. A double binds two hydrogen bonds with T, and C and G triple hydrogen bond together. Adenine and thymine bond with two hydrogen bonds, while guanine and cytosine bond with three. Due to the size of the bases, and the ability for bases to form hydrogen bonds is why that there is a difference between the two. But remember, the strands are complementary to one another. So CTG, CTA on one strand will be GAC, GAT on the other.